In today's webinar, Jason Hupp will be talking about complementary measurements using the LI-600 Porometer Fluorometer and the LI-6800 Portable Photosynthesis System. In addition to Jason, we also have a team of panelists. We have Brandon, who's with the sales group, Elizabeth, who is in technical support, and also Shannon Lorio, who's a principal scientist. I'm Abby Brooke. I'm a product manager here at LI-COR, and I'll be moderating today's session. So during today's presentation, if you have questions throughout, please send them to us via the Q&A tool in Zoom, and our panelists and also Jason will be able to chat you answers throughout the presentation. We actually had Jason record his presentation earlier, so I'll be playing that, but he'll be available to answer questions throughout. And then once the lecture is done, we should have some time to do some live Q&A. So we do have a large group attending today, so we're going to do our best to get all your questions answered. But if for some reason we don't, our team will be sure to follow up with you afterwards, and we'll also be sending you a link to the recording after today's event as well. So with that, I'll go ahead and get the lecture started.
Hello everyone. Today we're going to talk about the LI-600 and LI-6800, focusing on the types of things that the two instruments measure and the way and approach that they use to make those measurements. And I think as we do this, it's going to tell a nice story of sort of the way these two very similar yet very different instruments fit together. As we go through this, I'm going to hit on a number of the new features in the two instruments that I think will be interesting to a number of folks in the audience. As a starting point, I like to use this diagram to sort of facilitate this discussion. And I do this because I think for a lot of new users, particularly people that aren't or haven't been exposed to these instruments before, there's temptation to call something like the 6800 a photosynthesis meter. And I think that's kind of a gross oversimplification of what the instrument does and paints maybe a misleading picture, because it implies that the instrument's going to go measure one thing that represents photosynthesis. And I'm not going to go through the details of this diagram in any highly detailed way here, but I do want to point out that there's not one thing that we can measure here that represents photosynthesis. It's a whole collection of processes, exchanges of energy and mass that are happening at different scales inside the leaf that we collectively call photosynthesis or photochemistry. And we can't measure one point in this whole collection of stuff and really have any great detail of information about the whole of all of this, right? There's some things that are happening at the large scale, like on the left-hand side of the diagram here, processes that we call transpiration or assimilation, these diffusive fluxes of water vapor and carbon dioxide between the leaf and the atmosphere that are being driven by both sort of physical differences between the leaf and the atmosphere and are in some ways connected to biology, either by regulation of the diffusive pathway by changing stomatal aperture, or by demand for carbon through the carbon-Benson cycle. And then there are things that are happening at a very fine scale that are providing the energy flow through the system that is coupled to the things that are happening at the large scale, right? And that's like light capture at the individual photosystems and the way that light energy is being dissipated through the whole collection of stuff, whether it's being directed to these photochemical processes or it's being directed away from these photochemical processes. So in the instruments that we're going to talk about, we measure sort of 2 points that bracket everything here, right? So we measure gas exchange, and depending on the instrument, that is the exchange of water vapor or the exchange of water vapor and carbon dioxide between the leaf that we're measuring and the atmosphere. And we measure chlorophyll fluorescence, this energy dissipation pathway, specifically in our case, that's happening at photosystem 2, that gives us information about the state of the photosynthetic machinery and the way that energy is being used at that point in time. From fluorescence, this is not everything that we can measure in the instrument. Depending on the instrument, some measure less, some measure much more. But they're kind of the highlights of things that we might derive, and maybe a nice framework place to start in sort of thinking about, what we might do with these instruments. And so most fluorometers measure these things that we call quantum efficiencies, the most common of which many of you might recognize is FV over FM. The reason that this one's very common and most people recognize it is because evolutionarily it's a highly conserved value. If we measure happy, healthy plant materials, sort of regardless of taxa and location on the planet or anything like that, that value falls out at about 0.8. And as stresses that build in the leaf that physically degrade the photosynthetic machinery, like cold damage or heat damage, something like that, then we see FV over FM fall off. The next, maybe most common one would be this parameter called phi-PS2. It's the operating efficiency. It's how good the plant material is at that point in time. Capture and conversion of light energy to do photochemical work. It's some value lower than phi-PS2 because under most conditions, plants aren't operating at their maximum efficiency. From phi-PS2, we can derive electron transfer rate that gives us information about electron transport down the electron transport chain if we know something about light absorption at the wavelengths of light the leaf is being exposed to when we make the measurement. If we have measurements in the dark and the light, then we can make estimates of non-photochemical quenching, sort of this set of pathways that the plant uses to direct energy away from photochemical work. Again, if we have measurements in the light and the dark, then we can make some inference of the state of the reaction centers in photosystem 2, these parameters that we call QL or 1 minus QL, which are the fraction of open and closed reaction centers respectively. From gas exchange, not only do we get these diffusive fluxes, what we call apparent transpiration if we measure it in the chamber, and the carbon assimilation rate, which we might be able to label something like A net or A gross or dark respiration, depending on how we actually measure it in the chamber. But from transpiration, we can derive sort of our first pass at information about diffusive limitations, a parameter that we call stomatal conductance to water vapor that gives us information about the state of the stomatal apertures. I'll talk in some detail as we get through here as to sort of how we get from transpiration to stomatal conductance to water vapor, because this is one of the places where the instruments we're talking about really fit nicely together. If we have stomatal conductance to water vapor and the carbon assimilation rate, then we can estimate the stomatal conductance to CO2. And from that and the assimilation rate, we can make an estimate of CI, the pool of carbon in the intercellular airspace. Now, as is probably obvious from the title of the talk, we make two combined gas exchange and fluorescence instruments. They're the subject of what we're talking about today. And that's the LI-600 Porometer Fluorometer and the LI-6800 Portable Photosynthesis System. Now, the 600 is a small, handheld, portable fluorometer porometer. I show a picture of it here, clamped on a leaf, and a picture of it not clamped on a leaf, and we're going to call out sort of where various measurements happen in this system. So there's a small aperture in the middle of that white clip. That white clip opens up, we slide a leaf underneath it, and that presses a leaf down onto a steady state porometer that's measuring apparent transpiration and stomatal conductance to water vapor from the lower surface of the leaf. Downward looking on the opening in that aperture is an open fluorometer. It uses ambient light as the actinic light source. It includes a modulating beam and a saturating pulse, such that if we're in the dark, right, we're in a dark environment with the instrument, we can measure FV over FM. If we're in the light, as it's shown here in the image, then we can measure phi-PS2 with our PAR sensor on top of the instrument and some assumptions about light absorption. Then from that phi-PS2 value, the instrument calculates an electron transfer rate, and it reports that. The instrument also includes now, in the newest version, or the newest iteration of its hardware, a GPS, accelerometer, and magnetometer. So not only do we now get all of these physiological parameters, but we get a physical location where the measurement was made on the planet. And with the accelerometer magnetometer, in addition to the GPS, we can make some measurements that give us information about leaf angle and orientation. And from leaf angle and orientation, relative to the position of the sun, which we know because of the GPS location, we can provide estimates of fraction of direct light incident on the leaf. And so not only do we get information now that's physiological about the leaf from the porometry and the fluorometry, we get location, orientation, and information that provides contextual bounds around the environment that the leaf is seeing. The 6800 is a bit of a different beast, right? You can see it's not a single handheld instrument like the porometer, fluorometer, the LI-600, right? There's a reason that we call it a system. There's multiple components to it. The large white chunk with the handle on it that we see in the middle of the image, that's what we call the sensor head. The sensor head is where the leaf chamber is. So the arrow from my gas exchange call out here is pointing to where the leaf is clamped in the instrument. The chamber opens from the bottom. Both surfaces of the leaf are clamped into the chamber. Above the chamber is a light source. And in the case of the way the instrument's configured here, that light source is combined with a chlorophyll fluorometer. This is a full function fluorometer where we have control not only of actinic light intensity, right? That's the light source component of it. But we also, the way that we do the actual fluorescence measurement is a bit more full featured. We include some extra types of LEDs, particularly far red LEDs in the light source that allow us to do some extra measurements. And so not only can we get FV over FM, 5PS2, ETR, but we can get FV prime, F over FM prime, NPQ, we can get the QL parameters, amongst a whole host of other things. We also include a par out sensor on this instrument. The chunk in green setting on the tripod we call the console. The console provides the user interface, which you can't see here, but is a full color touchscreen on top of the instrument. It holds the batteries that power the system. It's where the data gets stored. And it includes all of the air conditioning infrastructure. So while our 600 was an ambient environment instrument, so it's measuring under the ambient light intensity, it's measuring at the ambient CO2 concentration, it's measuring at the ambient humidity. In the LI-6800, we control all of those things. So the console provides a controlled CO2 concentration at some user set value between 0 and a little over 2000 ppm. It provides a airstream at a fixed water vapor concentration, and we can control that concentration in whatever unit you prefer to think in. The right answer is VPD, but you can do what you like. And we include a temperature control module in the sensor head. So now we can control not only the light intensity the leaf is exposed to, we can control the CO2 concentration the leaf is exposed to, we control the VPD the leaf is exposed to, we control the leaf temperature itself, or the temperature of the air around the leaf, which is quite a bit different than what we do in the 600, right? We're going to come back to these differences in the actual measurement environment, because this is one of the places where we really see differences in how we apply the instruments. But before we get to that, I want to talk a little bit about these gas exchange measurements specifically. There's not really anything particularly exciting about the way the 6800 does it, but it provides a good framework for talking about the 600. So the 600 and the 6800 are both what we would call open flow through steady state gas exchange systems. And the way that the 600 measures apparent transpiration is the same way that the 6800 measures apparent transpiration, at least fundamentally, which also happens to be the same way that the 6800 measures carbon assimilation. And so you can envision a box that would be our leaf chamber. You know, in the case of the 6800, like I said, that box is enclosing both leaf surfaces. In the case of the 6800, that box is really just pressed to one of the leaf surfaces. So it's not quite how I've drawn it here, but conceptually, it'll be the same for the way that we talk about this. So we have this leaf in the box. If that leaf is happy and healthy and in the light, the stomata are open, and so it's releasing water vapor into the box. And at the same time, it's taking carbon dioxide out of the box. Now, we have a flow of air that is constantly entering that box and constantly exiting that box. And that flow enters at some molar concentration for water vapor, some molar concentration for carbon dioxide. Once it enters the box, it begins to interact with the leaf. It spends some period of time in the box, depending on the volume of the box and that flow rate, where it is modified by the leaf. And then it exits at some different concentration for carbon dioxide and water vapor. So if we know the number of molecules going in, the molar flow rate entering the box, and the number of molecules exiting the box, the molar flow rate going out, and the molar concentrations coming in and the molar concentrations going out, it's a simple mass balance in the difference between those two to get at the individual fluxes. And this is cool because the system is inherently at steady state, right? So long as our leaf is happy and healthy, we can leave it in the chamber for as long as we want to make these measurements. And there's nothing about the way that we're making the measurement implicitly that's going to change the thing that we're measuring, right? The system is at steady state. It is at a constant set of conditions. Now, like I said, for the 6800, well, maybe this isn't all that exciting. Pretty much every modern photosynthesis system, whether it's from us or some competitor of ours, uses an open flow through steady state approach to make this measurement. And there's very good physiological reasons for why they work that way. It, however, isn't the case for porometers, right? Outside of the 600, other porometers use a different technique to make this measurement. And there's two basic approaches to this. Well, there's three. I don't show all three of them in the diagrams here. But of these basic approaches, they're more or less similar, right? There is a box that you press to the leaf. That box is sealed, right? We don't have a steady flow of air and a steady state, a flow of air out, such that the conditions in the box are constant, right? In both cases, whether we're talking about a transient porometer or diffusion gradient porometer, there is a transient that's happening in water vapor that's part of the measurement. So in a transient porometer itself, it's a time derivative measurement of the change in humidity inside that box that's used to calculate the transpiration rate, which in turn is used to calculate conductance, which means that there is some physical minimum time that's required to make the measurement based on deriving a large enough change in humidity to be measurable. And then there's some physical limitation to how long the leaf can be in the box, because eventually you reach saturation and you can't make the measurement anymore. In the case of a diffusion gradient porometer, it's the same basic issues, right? Like there's some amount of time that it has to be there, and then it can't be there too long because then things come into equilibrium and it doesn't happen. In a diffusion gradient porometer, the difference is that instead of measuring a transient as a function of time, we're generally measuring 2 points in space away from the leaf and fitting those 2 points to Fick's law to try and solve for the stomatal conductance, right? And confusingly, a diffusion gradient porometer is often called a steady state porometer, but it's a really different use of the term steady state. When we say steady state at LI-COR, we mean open flow through steady state gas exchange. I said there was a third model here. There's a modification on a transient porometer that's a null balance approach that alleviates the saturation issue, but it doesn't fix all of the issues. Now, what are the issues here? Well, the issues are, in both cases, it takes some period of time to make that measurement. In both cases, we're allowing humidity inside the measurement system to increase significantly over ambient, such that when we remove the leaf from the chamber, we have to allow the chamber itself to re-equilibrate at ambient before we can make another measurement. And that might take maybe it takes tens of seconds to maybe a minute or so to make a measurement. And then it's going to take a minute or a couple of minutes for the chamber to recover back to ambient, right? Because of the steady state nature of the measurement that we make in the 600, we don't have this recovery period because the chamber itself is never moving very far from the ambient humidity, right? We're constantly flushing that chamber with ambient air. Because we're at the ambient environment to start with, this limits the opportunity for any sort of physiological response of the leaf, right? We're not really changing the environment the leaf is seeing. And the instrument itself is tailored to have a really short washout time. What I mean is that it uses that flushing flow rate is relatively high compared to the volume of the chamber exposed to the leaf, such that the residence time for air in it is very short. We're talking about washout times that are fractions of a second. And we use some very high precision sensors in this instrument that we grossly oversample, which limits instrument noise. And the combination of this short washout and the high precision sensors and the fact that we're not changing the environment the leaf is exposed to means that the response time for a measurement on the instrument is incredibly fast. And I show some data here for an artificial conductance standard, but if you put a leaf in here, you'd see basically the same thing. This is from a bigger data collection set where I wanted a conductance that was going to be constant over several hours. And time zero here is the moment that I clamped that standard in the chamber. And you can see that at about 3 seconds, we're stable, right? It takes about 3 seconds to make the measurement in this case. Now, practically speaking, we're often more in the 5 to less than 10-second range on the 600. It depends a bit on how big the conductance is. We're generally quicker at lower conductances and maybe a little bit slower at higher conductances, but we're still talking worst case sub 10-second range. One of the nice features of the LI-600 is that for many instruments, when to log the data is an empirical choice left up to the user. The user looks at data in real time and they says, oh, it's stable. Oh, it's not stable. I'm going to hit the log button when it is stable. We've done a really nice job of parameterizing what stability looks like in the LI-600, and we include what's called an auto mode. And in auto mode, the instrument knows when it's stable, it knows when it's not stable, it knows when there's a leaf in the chamber, it knows when there's not a leaf in the chamber, and it can handle the data recording for you. This is a nice feature. The nature of how fast the measurement is in the LI-600 is really the lead-in to sort of this first big dichotomy I want to put between the 600 and 6800. And this dichotomy is related to the way that we classify the measurements that we can make with these instruments. And so at sort of a gross pass, measurements from these instruments, they fall in really 2 categories, right? You're either making what we call a survey measurement, which is where we go out at some point in time and we try to measure the instrument or measure the plant material with the instrument under the environment that the plant is currently exposed to get a picture of what it is doing at that point in time, sort of what its realized behavior in the moment is. And because the LI-600 is so fast and because it's measuring under the conditions that the leaf is already exposed to, it's really optimized for these survey measurements. And I have some example data on the screen here. This is from a larger study looking at drought stress in some wild pepper accessions. And I'm going to talk more about this data in sort of a different context here in a minute, but we'll just start with this, right? So these are 2 diurnal curves from a drought stress experiment. One collected on a large collection of these accessions before a drought stress was applied, and one, several days after the cessation of irrigation. So after it has been water stressed. And each one of these time points, so each time point here, is well over 100 measurements, right? So there's over 30 different accessions that were used in this experiment. They were grown in replicate. And what we did was we took an LI-600 and we went through and we measured every single plant in the experiment at each one of these time points. And that whole measurement sequence, took 20 or 30 minutes to get through all of the plants because it's a whole lot of plants. But it was easy enough to be done by a single operator going through and doing these measurements. And I talk about the time here because this experiment was done in collaboration with some other researchers who set it up. And before we came with the 600 to participate in this, they were doing these measurements with a 6800 to measure stomatal conductance. And in a broader time period here than what we get 5 sort of discrete time point measurements with the 600 of everything, that's how long it was taking them to go through and get a single measurement on each plant in the experiment for stomatal conductance with the 6800. So that data, it was great to collect it, but it's really difficult to do anything with it, right? Because not only do you have these differences between accessions that are sort of compounded into things, but you have this time of day effect, such that you might be sampling one accession at a few different times during the day, or maybe the way the experiment's laid out, you're sampling them relatively close in time of day, and you're trying to compare that to something else collected at a different time of day. And it's difficult. The 600's fast, so we can tease out these time of day differences because we can get sample sizes or sampling schemes that actually allow us to characterize this. Now I'm going to come back and talk a little bit about the differences between the individual time series that I've seen here in a couple of slides. So what's the 6800 good for? Well, the 6800, certainly we can use it for survey measurements. It is inherently slower for survey measurements, and most of that slowness is not actually a product of the instrument. It's really a product of the operator. Because the 6800, no matter how you want to look at it, it's a contrived environment in the chamber. Like, you as a user are setting the environmental conditions. You set VPD, you set CO2 concentration, you set light intensity, you set temperature. And, I have several decades of experience making these measurements now, and I think I do an okay job of thinking like a plant. And I can certainly say, looking back on how I started this, I had no idea how a plant perceived the world in the beginning. And I think, that's true of most of us when we start these things. We think we know how plants work. And then we get out there and we start measuring them, and we realize the way the plant perceives the environment is very different than the way that we intuitively perceive the environment. And it's hard for us to make the gap as, some large, mobile, sentient mammal, to thinking about the world like, a more or less sessile creature without, any sort of way of comprehending the things that it experiences. So that's a knowledge gap on us as a user. But even once we get past that, we're still talking about, in the context of a survey measurement for conductance, a measurement that's on the order of a minute or more, and maybe with some extra time added to that in the way that we annotate it. So the real strength of the 6800, while we can do survey measurements with it, comes from the fact that we can control this environment and we can change it. And we can look at how the plant responds to these changes. And this is what we would call a response measurement. Now, the most intuitive sort of response measurement is the one where we hold sort of all of the environmental parameters but one constant, and we iterate over that one. That's like the CO2 response curve on the top here, or the light response curve on the bottom, where we hold everything constant except for CO2 or light, respectively. And we change either the CO2 concentration or the light intensity, and we look at how the plant responds. These curves are very commonly done because we can derive sort of limitations or capacities of the photosynthetic machinery by fitting them to sort of to specific models that give us different parameters, things like light saturated assimilation rate, or the dark respiration rate, or the light use efficiency, or quantum yield of carbon assimilation, or parameters like VC max, this maximum rate of carboxylation, or J max, the maximum rate of electron transport. Or if we take the curve out far enough, we can fit the CO2 response curve for triphosphate utilization, which gives us some information about phosphate status of the plant. So it's a different kind of information than what we get from the 600, even though for a number of these things, we're measuring similar parameters. So 2 instruments measure similar things, in some cases the same thing. They approach it differently with respect to the way they treat the environment the leaf is in. And so maybe it's obvious at this point, but they're not direct replacements for each other. I can't take an experiment that was designed for 6800 type data and just go use a 600 to collect it. Can't take, maybe I can take a 6800 and use it to go do surveys of stomatal conductance, but maybe that's not the best use of my time. But there's also this place where the two of them sort of fit together and one becomes informative to the other, such that, we can really either more efficiently collect the data we're interested in, or we can really improve the quality of the data that we're collecting. And so the next bit of this talk is I'm going to walk through some examples, starting with that pepper data that I showed a couple slides ago, and talk about, ways of thinking about how these fit together. And the first way that I'm going to talk about how they fit together is, what I call the screening and evaluation program here. And you can imagine, like, okay, so that pepper experiment, we've got 30 plus different accessions, each one of those things in replication. That's a relatively small experiment for these things, right? If this were a breeding program, we might have hundreds of breeding lines that we're trying to screen, in this case for drought response, right? And hundreds of lines being screened with a 6800, I mean, that's pretty challenging. We need some way to really delve in and find the ones that we're interested in. And this is where the speed of the measurement in the 600 is really wonderful, right? Like if we're talking a 5 to 10 second measurement, even 100 lines being measured is not an impractical ask for even a single operator with an instrument. And so we can use the 600 in this context to screen for these sort of broad patterns of physiological behavior, either things that are related to what we can see from the fluorescence signal or what we can see from the conductance signal. And then we can use that sort of screening of broader physiological patterns to pick out, either the plants that don't respond or the plants that do respond, these physiological outliers. And we can go back with the 6800 and taking these more detailed response measurements, really start to tease out, the physiological basis of what's happening here. We can evaluate the limitations and the underlying mechanisms. And so obviously this is what we did with that water stress experiment in peppers, right? So we had a 600 with a single operator. We had a couple of 6800s with a single operator. We took them to this experiment that was set up. We did these diurnal curves of stomatal conductance with the 600, measuring at sort of discrete time points throughout the day, looking at the physiology, the conductance physiology, as we went through the day to look at how stomatal conductance responds. And in the plots, these gray points, this is the average for everything that we measured at each one of these points. And then those error bars are standard deviations on that average. So they're really broad, right? Because they include all of these different accessions, which have all of these different capacities for, or different physiological capacities driven by differences in genetics and where they came from and where they're selected for, right? So they're big error bars. And then I've pulled out the data for two individual accessions that I'm calling insensitive and sensitive here, based on their patterns and conductance at sort of midday after experiencing water stress, right? And they behave more or less similar to sort of the mean of all of these things in an unstressed condition. And then once they see water stress, the insensitive one, is pretty indifferent to that water stress at midday. It's got higher conductance than the mean. And then that sensitive one is pretty shut down. Its conductance is pretty close to zero, and it's not changing much throughout the day. Well, that's cool. We can see that water stress exhibits some sort of stomatal response in these plants. But really the goal here is not to know, what they're doing with their stomata. It's like, well, if I want to evaluate a plant that's really resistant or not to water stress, I'm interested in what's really happening with carbon, right? How does that change in stomatal conductance and the way that the plant is regulating water affect that plant's capacity to take up carbon, which it's going to use to support growth, and ultimately, because we're talking about peppers, fruit production, the thing that we're actually interested in. So like I said, we did parallel measurements with the 6800 on these sensitive and insensitive varieties. And what we see from light response data, so we did triplicate light response curves. Over a period of time during the day, it took a couple hours, right? So we're running two instruments in parallel. each one of these curves is 45 minutes or so. And so can't get a huge sample size, but we can get some. And what we see from the light response data is these plants are the same. On its surface, that's not maybe all that interesting of a finding, right? Like they're doing the same amount of carbon in assimilation, pre-water stress, post-water stress. And the two varieties that we're looking at, or the two sessions that we're looking at, aren't behaving really any different from each other in terms of carbon or energy use. Okay. But if we think about that for a moment, that's kind of a surprising finding, right? Because our sensitive variety, we already showed from the diurnal conductance data, is shut down under water stress. The stomata are mostly closed. So we could look at the carbon assimilation response to light a little differently, and instead of plotting it as a function of light, we could plot it as a function of conductance. And this should linearize the curve, right? So carbon assimilation should be more or less a linear function of stomatal conductance. And the slope of this curve is the intrinsic water use efficiency, right? And we can see that, well, okay, maybe obviously because we have the porometry data to go with this, that water use efficiency for the two plants is pretty much the same in a pre-stress condition. Under water stress, both of them tighten up a bit, but the sensitive variety tightens up a lot and it moves towards a zero intercept. Interesting. Well, stomata are shut down. We can see that it has higher water use efficiency under drought. So does that mean that these plants have increased their capacity for carbon capture? Well, sure, we can evaluate that using CO2 response. And so we did CO2 response measurements on these plants, and we can see that, sure, there's a higher stomatal limitation in the sensitive variety, and the way that the sensitive variety is overcoming that limitation and making and maintaining its carbon uptake capacity despite its closed stomata is that it has greatly increased its capacity to take up carbon. We can see that by this much, much higher maximum sort of assimilation rate at high CO2 here than the insensitive variety. Now, these curves maybe look a little odd compared to curves that you've seen for CO2 response before, and that's because they're collected with a different technique than what would traditionally be used. This technique has been implemented on the instrument for about a year now. When we collected this data, it was before it was publicly released, so when we've talked about this data before, we've sort of omitted this detail. But the data collected here was collected using this dynamic assimilation technique, which is a re-evaluation of the way the mass balance is done on the LI-6800. You know, in the way that I showed it previously, you know, we make this assumption of steady state, that the incoming concentration is constant, right, at some fixed value, and the only thing driving changes in the system are what's happening inside the chamber. And because of that, you know, we can derive what's happening inside of the chamber. We've met the steady state assumption. The dynamic assimilation technique removes that requirement for the steady state assumption, right? It allows us to calculate the mass balance on top of transient conditions that are happening with the incoming concentration to the system. And so instead of making discrete measurements of carbon assimilation as I step through different CO2 concentrations here, what the DAT technique let us do was we could just slew the CO2 concentration. We could provide a continuous ramp in the concentration. And the instrument itself could solve for the actual assimilation rate on the fly as we went through that. It's not like previous non-steady state techniques where you required sort of an empty chamber calibration to correct for the time differential between reference and sample that's inherent in these instruments. No, this one is solved on the fly, and so the data that we get directly off of the instrument from the measurement we can use, which is pretty cool. It allowed us to do these really high density curves, but it also allowed us to do these curves pretty quickly, right? I think these were maybe 5 or 10 minute DAT curves that we did on the pepper, whereas a steady state curve would have probably taken us 45 minutes or maybe an hour, depending on how many data points we wanted on the curve. So that's sort of a screening evaluation approach to this. Another way that we might approach how these two instruments fit together is just thinking about like the data that we get from an instrument. And I think uh, many people, admittedly myself in the beginning, all those years ago, we think about these things as like a yardstick, right? We're going to walk out, we're going to put it on something, and we're going to get a number. And it's maybe not intuitive at first that those numbers aren't, direct measurements. Particularly in the context of the 6800, almost nothing we report on that instrument is a direct measurement, right? They're the product of some direct measurement, some calculation that requires some degree of user input or some set of instrument constants. I mean, even things that we think about being direct measurements, like often we'll call like the carbon dioxide concentration reported by the instrument a direct measurement. It's not. We're measuring the current, the electrical current output from some detector. That electrical current is related by some set of instrument constants to the gas density in the optical bench when we measured that current. Those instrument constants being the calibration coefficients. That gas density is related by the ideal gas law to the concentration. And you can suddenly see, even to get something as simple as the gas concentration that we start with for many things, like there's a whole host of calculations that are dependent on things to get there. So even if we call the gas concentration a direct measurement, which again, I've already said it's not, to get through to all of the things that we want from the instrument, there's still this cascade of calculations that have to happen. So if we start with gas concentrations and we want to calculate fluxes as, say, a first level output, well, we need not only the concentrations, but we need flow rates. We need some user inputs. We need to know how much leaf area is in the instrument. And that gets us our first level output. And then if we want to get to, say, the next level of output, say a gas conductance along the diffusive pathway, like stomatal conductance to water vapor, stomatal conductance to CO2, well, we need some more direct measurements, right? For stomatal conductance to water vapor, I'll show in a minute, we need leaf temperature, right? And we also need some more user inputs and some more instrument constants to get us there. And so I want to use the way that we get from transpiration to stomatal conductance to highlight a really, really neat use of the 600 that I think opens up a way to better constrain and improve the quality of our 6800 data that wasn't easy to do before. So let's talk through how we get stomatal conductance to water vapor. And so I'm back to my cartoon of a leaf here, and I've added some extra definition to it. And that extra definition is sort of parameterizing Fick's first law of diffusion for our model leaf, right? And Fick's first law of diffusion says that the diffusive flux of some component X, right, in this case it would be FW that we're interested in, is a function of the diffusive conductance along the diffusive pathway GTX for that gas species, times the difference in concentration between the endpoints of the diffusive pathway. And in the case of water vapor, those endpoints are really the surface of the mesophyll and the boundary between the boundary layer, this layer of still air built up at the leaf surface, and the well-mixed air outside of the boundary layer. So we can take this model and we can plug it in for actually water vapor, the way that we see things in the chamber, and you get something that looks like this, that says the total conductance to water vapor is a function of the transpiration rate that we measure in the chamber, and the difference between the water vapor concentration inside the leaf, which we get by assuming the leaf is at saturation at whatever the leaf temperature is, and the water vapor concentration in the air outside of the leaf, which is the water vapor concentration we measure in the chamber. So we can solve for GTW, which is total diffusive conductance. And if we think about what GTW is, it's a collection of a bunch of different conductances that some are under biological control, like that of the stomatal aperture, and some are just physical, like that of the boundary layer, right? And so we can borrow from Ohm's law here, and we can build a resistive model. We'll note that a diffusive resistance is the inverse of a diffusive conductance. So 1 over R equals G, and thus 1 over G equals R. And Ohm's law gives us sort of scaling rules for how these things sum together. And so we can define a diagram that says, We start with a resistance in the intercellular airspace, what we call Ri. That resistance feeds to two resistances that set in parallel to each other, what we call Rc, the cuticular resistance, and the stomatal resistance, Rs. Rs has an arrow across it because it's a variable resistance. And those feed into a resistance to diffusion across the boundary layer, what we call RBL. And if we write out the full model for this, it's fairly ridiculous because we can't solve it because we don't know everything we need to know, so we start making some simplifying assumptions. For water vapor, these simplifying assumptions are pretty reasonable, right? The first one is that the cuticular resistance is quite large. It's not infinite. There is some diffusion of water vapor across the cuticle in most plants, but generally the diffusion is small enough that it doesn't matter for most of the things that we want to do with these instruments. And by assuming that cuticular resistance is large, then we can assume that cuticular conductance, the 1 over Rc, approaches 0, right? So the inverse of infinity is 0, so the inverse of something large starts to approach 0, and we just ignore it in the calculation. And then the next assumption we make is that because the gradient in humidity inside the leaf is relatively small, we can assume that intercellular resistance is small, because even if intercellular resistance was large, because the gradient inside the leaf is small, we get to ignore it. And that leaves us a simplified model for separating the stomatal conductance from the total conductance, where we just need to know the total conductance to water vapor that we calculate from the apparent transpiration rate and something about the boundary layer. Now, my discussion here is not about the boundary layer per se. We know what boundary layer conductance is in the chambers pretty well. We spend a lot of time carefully characterizing this and ensuring that it is some rather large value in all of our chambers. That is a fairly significant part of the development effort for any chamber. The point of the discussion here is about that little 1 that sits on top of the boundary layer conductance in the model that I show here. This model is very much exactly as I've drawn it. It is a one-dimensional model representative of diffusion across a single plane. Now, think back to the way I described how the leaf sets in the two instruments, right? That's exactly how the leaf sets in the LI-600. It's a one-sided measurement, right? So we're just measuring stomatal conductance or apparent transpiration from the single leaf surface, which is as described by the model. In the 6800, however, we're enclosing both leaf surfaces, which means that the total conductance that we're measuring has to be partitioned across those surfaces. And the way we do that is by splitting the boundary layer using this factor that we call Kf, which is derived from the stomatal ratio. And the stomatal ratio is essentially the ratio of stomatal conductance on one side of the leaf to that on the other side of the leaf. Now, the instrument itself has no way of knowing this. This is a user input. It's something that you have to put in. And by default on the 6800, we set it to 0.5, which says that there are twice as many stomata on one surface of the leaf as the other. The direction of how that ratio is calculated doesn't matter. It doesn't matter if it's 2 times as many on the top or 2 times as many on the bottom. It's 0.5. It gives us the same thing. So it should be clear now, to get the best measure of stomatal conductance in the 6800, we need to tell the 6800 what our stomatal ratio is. So how do we get a stomatal ratio? Well, the traditional methodology would be to count stomata on both leaf surfaces using stomatal peels and assume that there's some fixed correlation between the stomatal density and the stomatal conductance. Maybe that's a good assumption, maybe it's not a good assumption, but either way, counting stomata from a stomatal peel is a slow and painful process. Because the 600 is a single-sided measurement, it brings a new approach to parameterizing stomatal ratio. That is, that we can walk out with the 600, and in 5 or 10 seconds, we can measure the stomatal conductance on the lower side of the leaf, and then we can flip the leaf over. In another 5 or 10 seconds, we can measure stomatal conductance on the top of the leaf, and we can take the ratio of those two, and we have the stomatal ratio to use for our measurements. Now, how much does it matter whether or not we do this stomatal ratio parameterization or not? Well, I have some example data from an experiment we participated in in hemp a couple years ago now, where we did exactly this, where we collected a bunch of data with the 6800 and we collected stomatal ratios with the 600 and we look at the effect. And this experiment was a really neat place to do this in. I mean, I think we kind of knew up front that it was going to tell an interesting story for this, but we didn't know maybe in the end how interesting it was going to be. So part of what makes this interesting is that this particular field experiment that we were participating in was on a commercial hemp variety. And this commercial hemp variety is produced by tissue culture, by cloning, right? So we have a field of genetically identical individuals. And just by the nature of the way the field was set up, there were 3 distinct planting dates for those individuals. And so on the same time in the experiment, under the same environmental conditions, we could measure plants that were in distinctly different phenological stages, but were genetically identical to each other. Right? So we're looking at phenological differences between them and really nothing else. And we collected a bunch of data as this, and the only data that I'm really going to talk about here is the CO2 response data. And so I show CO2 response curves here for the sort of 3 different phenological stages that we were looking at. And so the youngest plants, the most recently planted ones, were purely vegetative. They had yet to begin initiating inflorescences. These sort of middle-aged plants that we were looking at were at the point where they were just starting to initiate inflorescence development, but they hadn't started actually flowering yet. They weren't developing flowers. And then the latest stage plants were in full-on floral development, right? So they're at their flowering point in their life cycle. So 3 distinct phenological stages. We see some differences in physiological behavior in the CO2 response curve. Not really the point of the discussion here. The point of the discussion here is that to get that CI, as I mentioned earlier, we need to know stomatal conductance to water vapor. And to get the right stomatal conductance to water vapor, we need to know something about stomatal ratio of the leaves that we're measuring. So what did the 600 tell us about stomatal ratio in this case? Well, it told us that the stomatal ratio in this particular hemp cultivar was somewhere between 5 and 10 times smaller than what the default value on the instrument was, meaning that it was somewhere between 0.1 and 0.05. Right? It was 0.05 in the oldest plants undergoing floral development, and it was about 0.1 in the youngest plants that were still purely vegetative. And that stomatal ratio was different with phenological stage, right? So now let's think about what this implies. Well, it implies that using the default value adds some error to these measurements. That error scales with phenological stage, so it's not just a fixed bias in the measurement. And because of the way that stomatal ratio is applied in the calculation of stomatal conductance, that error is not only a fixed scalar at each vegetative or each phenological stage, but it is a function of the conductance that we're actually measuring. And so you can see in the plot here, I have the actual stomatal conductance corrected with the right stomatal ratio, plotted against what I call fraction measured. So that's just the ratio of that actual value to what the instrument reported with that default value of 0.5. And in all cases, the actual GSW is larger than the value reported with the default value on the instrument. The degree by which it is larger is different for each phenological stage because the stomatal ratios are so different from each other. And the degree of the error scales linearly with the stomatal conductance that we're measuring. So at the best case, at the lowest end of the curves, we're talking about errors that are a couple of percent. At the worst case, at the high end of the curve, we're talking about errors that are in the 10 to 12% range. That's pretty significant, right? We probably care about that magnitude of error in these measurements, particularly for making direct comparisons between these. Now, I said this is for ACI data, like CO2 response data, and we've talked about how stomatal conductance goes into the CI calculation. Well, these errors don't scale linearly into CI. What might be a 10% error in stomatal conductance here generally is going to be something less than like a 1% error in the actual CI value. So the error from using the wrong or the right stomatal ratio has very little impact on the absolute value for CI. The place where it gets us in the measurement of the ACI response is that stomatal conductance is not constant across the curve. And so while it's a small absolute error, the error is changing as we go through the measurement, and thus it means that it has a differentially larger impact on parameter estimates. So in the case of the data here, when I go back and fit the curves with the default stomatal ratio versus the curves that have been corrected using the actual stomatal ratios that we measure, what I find is that VC max is the most sensitive parameter to this. And in the best case scenario, we're talking about errors that are less than 1% differences in VC max between those. And in the worst case scenarios, for the vegetative plants, for the floral development plants where the stomatal ratio is so low, those errors in VC max are, between 5 and 10%. They're much larger. So if you want more details on sort of the two experiments that I talk about here, the things that the instruments measure and sort of how they fit together, we have a really nice application note that summarizes all of this in much greater detail than I've gone into here. And you can pull this from our website. There might even be a link to it in the follow-up emails that we send related to the webinar. But moving forward from that, I want to talk a little bit about some other sort of distinguishing things between the instruments, and this will give us an opportunity to talk about some of the other new features between them. So I want to say that the 600 is really a single purpose instrument. That's probably obvious from what we've talked about now, right? Like, it is an ambient environment instrument for surveying these particular physiological parameters, but even more so than that, because of the way it seals to the leaf, it's really purpose-built for broad leaves that are greater than 7 millimeters wide. That's the aperture of the chamber. And we've done a lot on the instrument to really optimize it for high throughput measurements, right? One of the newer features in the instrument is that, we've reworked some of the workflow to reduce the number of button presses. I can tell you my colleague that collected the conductance measurements on the peppers that I showed, he was doing, to get that data collection at sort of the volume that he was doing using the initial workflow on the instrument, that was hundreds upon hundreds of button presses in a day. I think for the way it was set up then, it was 3 button presses, including the way that we were using the barcode scanner for each one of those measurements. So a day worth of measurements was like 1,500 button presses just to collect the data. And I mentioned the barcode here because since we've released the instrument, it's always had the physical hardware to read barcodes. And that's why I picked the picture that I show here with a barcode hanging in front of these plants, because a barcode is a really efficient way to encode information with the plant and then to read that barcode and encode that information as metadata with the measurement. What we've done that's sort of new in improving this process, not only in reducing the number of button presses, but we also now include a barcode generator in the software for the 600, where you can take a list of barcodes, say a CSV file that you've exported from Excel that has all of your different plant identifiers or treatment identifiers or whatever, and upload it into the 600 software. And from the 600 software, you can print a series of code 128 or QR code barcodes that you can use to tag your plants and make measurements of stomatal conductance, and then scan that barcode to encode that information with the data. It should be, or maybe it's not obvious at this point, but it should be obvious that the 6800 is not really a purpose-built instrument. It is a platform, a gas exchange platform, that is highly adaptable to different sort of plant geometries and different sorts of sample materials. And so I show sort of an array of chambers that we've had available for the 6800 for a while here. These are things for measuring leaves of different sizes. So, the 3x3 leaf chamber, the 6x6, the large leaf chamber that we can adapt for needle measurements. We have chambers that we use to measure discrete samples, like the bryophyte chamber at the bottom, or small whole plants like the Arabidopsis chamber above it. We have different types of light sources for it. We have a soil chamber. We have platforms that allow people to build their own custom chambers and interface them to the instrument. The sky is pretty much the limit what you can measure here. But traditionally, that limit in the sky has been measuring things in air. And the really new feature of the 6800 in regards to chambers is the addition of the 6818 Aquatic Chamber. So now, not only measuring samples in air with the instrument, but you can measure samples in liquid suspension. And I showed the chamber fitted to the instrument here with an algal suspension in the sample volume. And this chamber pairs onto the fluorometer for the 6800 and allows us to measure carbon assimilation and chlorophyll fluorescence from a sample in liquid suspension. And the thing that's really neat about this, I think for people that are experienced at doing leaf level physiological measurements, this isn't going to sound all that exciting because we always use a steady state open flow through approach for those measurements. We've done that since sometime in probably the late 80s. And that's just the standard method for it. But this is quite a new thing for measurements in liquid suspension. So traditional measurements in liquid suspension are time derivative transient measurements, and they're typically done with oxygen, right? The carbonate system in water is rather complex, so it's difficult to measure a CO2 concentration. The way oxygen is very easy to measure in solution, but oxygen sensors themselves are generally low resolution. So the way most people make these measurements is they seal up a little bit of a sample and they measure the rate of change in oxygen generally over several percentages to tens of percentages to derive the oxygen evolution rate, which they relate to photosynthesis. Here, we use our standard steady state approach to measuring carbon. We measure tiny differentials in carbon across the liquid sample. And we do that under steady state conditions such that we can leave the sample in this fixed environment for as long as we want. We're not going to have any feedback. The oxygen concentration isn't changing. It's a very new way of approaching this measurement. And the thing that really makes this possible is the way that we equilibrate the water vapor concentration coming out of the sample chamber with the water vapor concentration elsewhere in the system. So there's a device, we can't see it in this picture, it's on the back side of the black chunk of the chamber that we're looking at the top, that we call the water vapor equilibrator. And what that does is it prevents condensation, liquid water, from making it downstream in the system and damaging the stuff that we don't want water to damage. And it reduces an inherent measurement uncertainty that happens when we try to measure very tiny changes in carbon on a background of a very large differential in water. Now, the aquatic chamber was first built and developed to measure algal suspensions as shown here. But with it, we also have this sample adapter kit that allows us to adapt the chamber to measure discrete samples in a liquid background, right? So instead of individual cells floating around in liquid suspension, we can measure a chunk of something, a chunk of plant material put in the chamber in some measurement solution. And you can see here, the window on the side of the chamber comes off. We put this adapter ring in place. With the adapter ring, we give you a plug that seals into that. The plug has some threaded holes in it. It has a little well. You can adapt it to your particular sample material. I show it here with some standoffs that are holding a piece of sargassum, marine algal, marine macrophyte. We would slide that plug into the side of the chamber. The plug in the top of the chamber would come off. We'd fill with our measurement solution. We'd close it up, and then we could make the measurement we want to make. We've done measurements of marine macrophytes in it and freshwater plants. We've had collaborators that have done measurements of marine macrophytes. We've seen it used for measurements of carbon assimilation by corals in the chamber. And then it also works really well for measurements of things that aren't in liquid suspension, but things that are saturated. And so you can imagine something like sphagnum moss, where its physiological activity is closely tied to the water status of the plant material, right? It's basically a sponge, and when it dries out, it shuts down. And traditionally, if we put that sponge in a chamber, it was dumping a ton of water into the chamber. It was drying out during the process. And the physiology that we were interested in was sort of shutting down as the process of us trying to measure it. And it was a pain. When we put it in this chamber, because of the way the equilibrator works, we end up recycling a lot of water vapor between the exhaust of the chamber and what air gets returned to the chamber, such that the equilibrator itself actually minimizes the evaporation rate from the sample. So not only can we leave a liquid sample in the chamber for a really long time without evaporating it, but we can leave a wet sample, something like a moss, in the chamber for an extended period of time without drying it out. So this is pretty cool. And this is maybe the biggest, newest change to the LI-6800 to talk about. And I think it concludes the things that I'm going to talk about today. We'll leave the chat open here so that if we have any questions, we can answer those now. And I'll say thank you, and thanks for listening.
Okay, like Jason mentioned, we will stick around for a while and answer additional questions you have. Go ahead and keep typing them in to the Q&A. As a reminder, we will be sending you an email with a recording to this presentation, as well as links to the app note and some comparison tables that we've put together. So you'll have access to those, and we'll also include some recent LI-600 publications and references. We've put a list together, and I know we've had some questions regarding if we had any of new references available. So we will include that in the follow-up email as well. I will go ahead and keep this webinar open for probably another 5 minutes. So if you do have questions, by all means, type them in, or if you think of something later on, feel free to shoot us an email. Jason, there's a question for you, actually, that Mei Ling just asked. She's wondering if it's possible to add the equation in the LI-600 data file Excel, just like the data from the LI-6800, so users can recompute some of the parameters if needed.
Hi, Mei Ling. Well, we've certainly talked about it, and I think it's not impossible that it is something that you would see from us in the future, but I can't say when or if it will happen.
Okay. Okay. There's another question that came in, and whoever wants to unmute. In order to choose the method to set the stomata ratio, is there's a large difference between stomata number and the stomatal conductance? Should I always use the stomatal conductance to set such ratio?
I can answer that one. Well, that ratio is actually the ratio of conductances. Previously, it would get referred to as just stomatal ratio, and we'd imply density as the way to work it out, because there wasn't really any better way to do it. I guess even back with the 6400, we had some discussion of how to do single-sided measurements to parameterize this, but I don't think many people did it because it was quite difficult. The idea that density is related to conductance in such a way that yeah, I don't know that it's a very well tested assumption that density correlates directly with conductance. So if you can use conductances, that's a way better way to do it.
Okay, thank you. All right. Well, again, thank you all for joining us. I'll keep it open just a couple more minutes. I'll turn some music back on, but keep sending your questions in and we'll go ahead and type up some answers. Thank you.